Hello students, we're going to be talking about the flow of genetic information in the cell and going through a brief overview of what's known as the central dogma, which is the idea that DNA is transcribed through transcription into messenger RNA that's translated into protein. We will also be briefly discussing DNA replication. I want to tell you that this has a lot more complexity than I'm going to be presenting. This is just a brief overview and not in the details that we've done our other chapters, but it is just trying to get you to understand the relationship between DNA and protein, that the DNA codes ultimately for all of those proteins that we've seen throughout the whole semester, the transport proteins, the enzymes. So our class has focused so much on protein that it would be odd not to mention how proteins are made. Okay, so before we do that, we're going to talk about DNA replication. So when you talk about double-stranded DNA, there has to be a way, first of all, of unwinding it and separating the two strands to replicate. The unwound portions can be susceptible to nucleases that'll attack single-stranded DNA and degrade it. The other thing is that your two strands are anti-parallel. You have one going 5' to 3', the other 3' to 5'. So anything that's going to try to duplicate those strands has to take that into account. And then there has to be a protection against from errors in replication. Okay, so this is the idea that if you have two parental strands, that each of the strands they'll separate from each other, and then you'll create two new strands. The replication is known as semiconservative, meaning that each strand serves as a template for a new strand. And so when you're done with the replication, you will end up with a strand with its copy and another parent strand with the copy. In DNA replication, the DNA double helix has to unwind, and that point where it's unwound that allows for replication is known as an origin of replication. In eukaryotic cells, there tends to be, in DNA replication, more than one origin. Okay, so here's that statement, eukaryotic cells, eukaryotic DNA. There are several origins of replication. Okay, so this is a simplified diagram, but it's really the heart of what I'd like you to understand about it, which is that the two strands have to be, they'll unwind, and there is the 5' to 3' parental strand, which is here, and then the 3' to 5' parental strand, which I've circled up there at the top. So with this in mind, what will happen is that when the 3' to 5' strand is our template, it's easy for the polymerase to go ahead and make a copy because the polymerase naturally wants to go in a 5' to 3' direction. Where it's unwound here, this is called the replication fork. Okay. All right. So it's going to go from a 5' to 3' direction. Now, the other strand is more problematic because if you notice, since the parental strand is 5' to 3', the polymerase can't go in that same way. It has to go the other way. So it's going to have to fill in this direction. And it does so in a semi-discontinuous way, meaning that it creates fragments, and these are called Okazaki fragments. They are 5' to 3', but then you have that as fragmented, and that has to be, those gaps have to be filled in by a special enzyme called DNA ligase. Okay, so just to recap again, since the DNA polymerase shown here in the green has to move in a 5' to 3' fashion, it can move along the parent strand that's 3' to 5' just fine. It's going to go, and that's going to be called the leading strand. And it's going to be formed continuously. In the other strand, it's going to make a copy, but it's going to continue to move 5' to 3', but you notice it's this other direction. And then you end up with those Okazaki fragments that have to be filled in with DNA ligase to complete the copy. Okay. Now with transcription, what transcription is it's going to take your DNA as a template, and an RNA polymerase is going to be used to make messenger RNA. So this process of transcription is DNA forming messenger RNA. Okay, this is done by an RNA polymerase enzyme. And well, if you think about it, we're going to need the nucleotides, right? The nitrogenous bases, the phosphates, right? The backbone has to be made there. So with this in mind, we will be using, or it will be using RNA polymerase. For adenine, it's going to use ATP. For guanine, GTP, cytosine, CTP, and then for the uracil, it's going to use UTP. And if you think back, if you can't quite see this, there was a picture in the previous video lecture that showed ATP. It had adenine attached to a sugar with 3 phosphates. And so you can kind of get a feel then for how that's going to be used to build the mRNA chain. The RNA chain is synthesized in the 5' to 3' direction. Okay, and the nucleotide at the 5' end will still have its triphosphate. Okay, when this occurs in transcription, the DNA template is not changed in any way. Okay, all right, so let's go to the next picture. In this case, the RNA polymerase is shown as the brown circle here, the enzyme. And the DNA has what is known as the coding strand. That's up here, the 5' to 3'. And the coding strand is called the nontemplate or the sense strand. And then the 3' to 5', that's your template. That's going to serve as the template strand because the RNA polymerase, kind of going back to here, let's go back this direction. The RNA polymerase synthesizes in 5' to 3' direction. So it's going to use the 3' to 5' as the template strand. Okay, so the polymerase then will create it's not a copy, but it's the messenger RNA. If you notice, let's take a look at the code. G on the template strand would produce a C on the RNA strand, messenger RNA. The A would go with a U. The G would go with a C. The C would go with a G. The A with the U. It's kind of hard to see now. We're getting a little bit further out. So I think the one I skipped was this T would go with this A. So they do have to complement each other. So kind of just writing the green part out, right? The template strand is T, C, G, A, G. So if you notice, the messenger RNA is C, U, C, G, A. So this was the DNA, and this is the messenger RNA. And notice again, the messenger RNA has uracil and not thymine. This is kind of a space-fill type model for the RNA polymerase. It is a very large enzyme, and it is an interesting, it's just to kind of give you a feel for how large and complicated of a structure it is, but I won't ask questions about that part. And this kind of gives you a feel for the inside of, this one is a yeast RNA polymerase 2. And it shows some common features. If you notice, there has to be room for the DNA to come in there, has to allow for the polymerase to come in. And if you notice here, there's a kind of what you'd call, what they call a rudder. It allows for the template DNA strand to kind of come into position for the RNA polymerase then to start creating the messenger RNA. And then the last part that I think is so fascinating is the part where that messenger RNA is translated into protein. So this has a lot going on with it, where the process to synthesize proteins is in the ribosome. And the tRNA is involved as well, because the tRNA I think of as kind of the bridge between the amino acid and the messenger RNA. So the messenger RNA is read 5' to 3', and the polypeptide that's formed from it is synthesized from the N terminus to the C terminus. Okay, so let's go through the steps. The first step is that the tRNA that has an amino acid on it enters into the ribosome in addition to the messenger RNA that's there, and they have to complement each other. Okay, so the tRNA, we'll see it in a second here, has a region that will align with the messenger RNA in what's called a codon that has 3 letter designations. So let me give you an example here. The messenger RNA starts off with AUG. Okay, that is the code for the amino acid methionine. And so the transfer RNA comes in, and it's the transfer RNA that has that match to it that on the other side of it has the methionine. So the tRNA binds to the ribosome one by one, matching their anticodons, which are this message here, that matches the messenger RNA. The codon there then, AUG, is the code ultimately for the methionine. The next one, GUU, the transfer RNA comes in with the anticodon that matches that, and that has on the other side of the tRNA has the amino acid valine, because GUU is for valine. Then there's GGC, that's glycine, GAU, aspartate, AGU, serine. Okay, we can keep going and going. So this is such a fascinating process. So step 1, the tRNAs bind to the ribosome one by one. They match their anticodons, which is the bottom part of the tRNA that will align with the messenger RNA codon. The polypeptide chain starts to grow, and there's a peptide bond formed between, in this example, methionine and valine. Okay, this is done by transferring. There's a transfer from the first tRNA to the incoming one. Okay, so they form a peptide linkage. The first tRNA then is released, right? Do you see how it's gone? And now the methionine is on the valine strand. Then the next one comes in, okay? And so it keeps getting passed on down the chain. So the first tRNA is released, the ribosome moves one codon length along the message, allowing the next tRNA to come into place. And then the ribosome will eventually encounter what we call a stop codon. The stop codon is the signal UGA. That means stop, and then that's the point at which the polypeptide chain is released. So all the proteins that we've talked about in this course are made from messenger RNA that codes for them. And then all the amazing things that we've talked about, how the protein folds into one shape that gives it its structure and its function, all that happens after it's been synthesized. So here's tRNA, which is such an amazing molecule structure. If you notice, here's the messenger RNA, GUC, okay? That's the messenger RNA. And then the anticodon is on the tRNA. Notice those have to match. It's harder to see in the other one. So this would be CAG. Okay, and then this would code for a specific amino acid that would be attached to the other side of the tRNA. In this case, this should be leucine. Okay, so this picture here is really the meaning of our life, meaning of life on Earth, because this is the genetic code that codes for all the proteins in our body. So AUG, that wasn't a random choice for that example. That's what we call the start codon, okay? And that codes for methionine. And that's the starting amino acid. That's the start codon in eukaryotes. And then there are the stop codons. They're also called nonsense codons because they don't code. They'll allow for the amino acid to process to lengthen the chain to stop. Okay. All right. Let's look just kind of at some really interesting look up here. If you notice, I'll highlight it with the highlighter. These are the 3-letter codes for phenylalanine. These are the ones for leucine. Here's some more for leucine, proline, serine, tyrosine, histidine, arginine, methionine, there's start, valine. Okay, so you're getting the point that each of these code for amino acids. So CCU would be proline, ACU, threonine, okay? And so the other part to consider is that, let's say, there is some room a little bit for error, because if you notice that it's not so tight that only one codon codes for one amino acid, there's some flexibility. So you see how CCU is proline, CCC is proline, CCA is proline, CCG is proline. So if you notice that last base, there's flexibility there. Notice in all of them, the last base is more flexible. Okay. Or sorry, for those two would be histidine. These two are a different amino acid. So this is really the meaning of life, right? This is what our genetic code from DNA translated into messenger RNA, and then the messenger RNA can code for our proteins. All right, lastly, in summary, all 64 codons have assigned meanings. 61 of them code for amino acids. There's 3 that serve just to stop the process. Some, like methionine, have only 1 codon each. The third base is not relevant for some of them, as I highlighted earlier. The second base is important for the type of amino acid. If the second base is a U, the amino acids are coded for are hydrophobic. And then there are 15 amino acids coded by 2, 3, or 4 triplets. It's only the third letter of the codon that varies. And so this allows for, honestly, if there's some flexibility in case there's an error there, right? If there's an error in the message, it can allow for the same amino acid to be placed on the protein. The other part, though, is the idea, let's go back, of mutations, that if there's a mutation on a DNA level, then the genetic code might end up being different. Sometimes a different code on that codon may not have an effect because, let's say, for example, you're here. If the change is UCU and it changes to UCC, you still have serine, so that's okay. Where things would be different and more detrimental would be here, UCU is serine, but over here, the second one gets changed to a U. Now you went from serine that's polar to phenylalanine that's nonpolar, bulky or aromatic. That could be very detrimental to proteins, right? We've spent so much time talking about their properties. And so a change from a polar to a nonpolar could be very damaging to that protein. So again, to recap, that the third base pair changing here keeps it at serine, but the second one changes it to a different amino acid, in this case from serine to phenylalanine, and that could be a very, very detrimental mutation then. Imagine how important serine was to our serine protease, and now you've changed it to phenylalanine. That will definitely make for a non-functioning protein. We don't have an opportunity to talk more about those mutations, but some diseases are caused by a single mutation and change from one amino acid to another. We saw that with the sickle cell anemia example.